Hi, I'm Justin O'Sullivan, and I'm here today to talk to you about what my research group has been doing. In particular, what they've been doing in relation to how the organisation, the spatial organisation of DNA and epigenetic marks affects how genes are regulated and turned on and turned off, in very simple terms. So imagine, if you will, that this bit of Play-Doh here is actually DNA. Now, of course, the DNA is not a fat little ball like this. The DNA is a long, linear double helix, and I'll just demonstrate that with this piece of Play-Doh here. And so this represents the chromosome, and it's got genes that are arranged in linear order along it. Now, obviously, this is quite a long molecule, and the DNA inside your cells is approximately 2 meters long if you extract the DNA from one cell and you line it up end to end. Now, that DNA has to fit inside a small cell, and let me represent that with this bag. Now, if I try and put this in this bag, it doesn't fit, unless we fold it and bend it and twist it. And so it folds and bends and twists to get inside here. Now, that's really interesting for one chromosome. But your cells have more than one chromosome. You have multiple chromosomes. So let's take another chromosome here, a yellow one, and let's stick it in there. Now, the interesting thing about that is, again, it twists and folds and it contacts the other chromosome. And so the interesting thing that we've been trying to work on is how these contacts and how this folding within this confined space affects how genes are turned on and off and how they respond to their environment. Now, responses to the environment are important, and let's just talk about that for a second, because part of the response to the environment is to do with epigenetics. So what is epigenetics? Well, epigenetics are modifications that occur on the surface of the DNA molecule. They don't change the sequence of the bases in the DNA, that's the A, T, C, and G. Rather, you have things like methylation, which I'll represent with this blue mark. And it binds onto the surface of the DNA here. And that then turns the genes next to it off, in really simple terms. And you can have multiple marks like that along the chromosome. Okay? But there are also other types of epigenetic marks, which I'll represent, and I'll show them on a different chromosome. Okay? But this green represents another mark. Okay? And that also works to control the genes that are next to it. Now, the question that my group has had is, when you fold the DNA, do these marks work individually? Do they come together? Do the genes that are turned on work individually, or do they come together in space? And so the way the DNA folds is quite interesting. It folds up. We don't know all of the rules for this yet, but it all folds up. And when it folds up, things come together, various bits and pieces come together, and so we get a nice structure, something like this, which will fit now inside this bag very easily. Okay? Now, here we have things like telomeres that cluster, and we know this is important, and telomeres are really important for aging and things like that, so we've got the telomeres at ends of the chromosomes clustering. But what about those epigenetic marks that I showed you? Now, they were all separated linearly before. What's happened to them now? We think that they're working together, that they're creating an environment. And so if I cut through this nucleus, what should happen is you'll see when we open up the DNA that the epigenetic marks have come together here and here, and they've formed a spatial environment. And it's a spatial environment that's important because it's contacting not just the 3 positions on the chromosomes I showed you before, but now, because of the way the chromosomes are folded up, we have this gene contacting here, this gene is contacting here, this gene here, this one here, this one here, the gene here, 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 etc. So you're getting multiple genes contacting this spatial environment. And it's the spatial environment that's important because it's the spatial environment within which regulatory factors, things that turn genes on and off, the proteins that are involved, the machinery that reads your genes, actually work. And so this is what we think is really important, the spatial organisation of the DNA and the modifications that are actually on top of it. And that's what we've been working to try and understand.